Hi, I'm Annalisa Paul from the University of Florida, and I'm here to talk to you today about what plant molecular biology in space might look like in 10 years.
So why is it that we need plants? Why, as space biologists, are plants important to us? Well, pretty much it's because plants are what allow us to be explorers. We need plants if we're going to travel anywhere past the limits of a picnic basket. And because we're travelers and explorers out there, we need to understand how plants respond to spaceflight. So what do we know about them? They do indeed respond. And when plants are in a microgravity environment or in alternative environments, they know they're there. And they show us that they understand by changes in their transcriptomes, in their epigenome, their proteome. They change the way their metabolic processes work in order to adjust their metabolism to the environment in which they're sitting. We need to understand what's the metabolic cost of those changes, and furthermore, how we can help them mitigate those costs in order to better adapt to our needs and their health. In the past decade, we in the plant space community have independently identified dozens of different classes of genes that are important to spaceflight adaptation. To look at a big handful of these is everything from reactive oxygen species signalings and other kind of stress responses to the things you might think about, like plant hormones and gravity sensing. But there's other interesting things that we've come across as a unit is that things like light signaling have become really important. When you don't have gravity as a stimulus, sometimes you use light or other tools to help you navigate. Cell wall remodeling is a big category for pretty much everybody. Changes in the cell wall composition is paramount to almost any kind of changes that we see in spaceflight. Heat shock genes, calcium signaling, all the things that tell plants that something is wrong with their environment, or at least something is different in their environment, and here's how they're going to address how to fix that. Sometimes those responses are inappropriate. They don't really need to do X, Y, or Z kinds of things to be healthier, but it's a novel environment. And so they don't know what tool to pull out of their toolbox to help them adjust. So they throw out all sorts of things. Lots of different genes and lots of different categories. But bottom line is that plants know that they're in a new and novel environment, and they work hard to get them in a place where they're comfortable. The other things that we've learned recently is that there are epigenomic changes that are induced by spaceflight. And so in addition to the kind of primary transcriptome that we take a look at, we've also seen that there are modifications to the genome that then have an impact on how the transcriptome and proteome looks downstream. So you can think about it from this way, is that there's a physiological adaptation to spaceflight that's really nuanced by the epigenomic modification. Now, why is this really exciting? Primarily because it's possible that these modifications could be inherited to the next generation, and this has incredible implications for spaceflight exploration. So in other words, on Earth there's tons of epigenetic changes that are in eukaryotic genomes that play a role in environmental responses. One of the classics that has been explored in plants is that pathogen responses can be inherited to the next generation in a population that has been exposed. One type of epigenomic modification that's been strongly linked to these kinds of responses, for instance in the pathogen response that I just mentioned, is DNA methylation patterns. And these are the kinds that we have seen so far in response to spaceflight. Now in some cases, those environmentally acquired DNA methylation patterns can be transferred to the next generation, and there they can contribute to the ability of the progeny to cope with that stress that was experienced by the parental generation. How cool is that? A quote that you see often in books that talk about epigenomics and epigenetics is that epigenetics has transformed the way we think about genomes, and it's true. And it's exciting to see this being applied to the spaceflight environments. So what if genome modifications that have been accumulated by different generations in space are inherited? Well, these are experiments that are ongoing right now. So we'll know that first primary answer in a couple of years. But what about in the future? How will we use all of these data, the transcriptomes, proteomes, epigenomes, in the coming decades? We need to gain a bigger and more complete understanding of what these metabolic tools are that are important to the physiological adaptation of plants in space. And this in turn can then guide genetic engineering and genomic strategies that help us develop spaceflight adapted lines of crops and other kind of advanced life support plants. We know what the cells do, we know what is important to them, and we can take that information to make them better, make them happier. So where will we be in 10 years? I want to set the stage first just a little bit. Regard our lowly mustard plant, a relative of Arabidopsis, but it's also a relative of all these things. And in fact, it is all these things. So in the past, let's say, 1,000 years, humanity has been slowly modifying this lowly mustard plant into all sorts of different things that we like to eat. Cauliflower, broccoli, kale, cabbage, turnips, rutabagas. All these things are essentially mustard, same genus and species. So plants is a great example of how plastic they are and how we can modify them. So let's take it a step further, is where we'll be in 10 years from the perspective of engineering with all this information of what genes are important to that metabolic toolbox in plants. We can do all these things. We can modify the developmental pathways that redirect resources to make a better edible product and a less stressed plant. Changing the metabolic processes that tell a plant that it thinks it's under stress when in fact it's not. We can modify the cell wall constituents that enables plants to optimize that carbon partitioning for edible biomass and doesn't waste it in places where it doesn't need. You don't need a big stiff stem in space if there's no gravity to pull you down. Also think about the problems that we have in spaceflight with managing fluids on orbit with no microgravity, or excuse me, without gravity. And we can engineer routes that are more resistant to some of that water stress. So where will this take us? Well, we'd like to think that in addition to now starting out with a better modified plants from the genetic point of view with new genes and new processes, also think about the generations of plants that are now going to have these modified plants that will be growing on orbit. In addition to the things that we have entered into them, plants can help themselves. And these epigenomic responses can be true epigenetic ones. And the adaptations that they incur while they're trying to physiologically adapt to spaceflight environment can in turn result into inheritable processes that make a better adapted progeny. What about in 10 plus years? Where are we going to be then? We can take these steps one step further again and modify the developmental pathways, modify the metabolisms to make nutraceuticals, modify cell wall components to make bigger leaves that expand more and have more nutrition, roots that also have modified starch storage. So at the end of the day, what we have is a space plant that looks more like this. We've got something where you can eat every single part, and you can engineer it to give you different things in response to different light wavelengths and to different habitats, and allow us to use and maximize the resources that we have available to us by engineering the plants as well as engineering the habitats. So where will we be in 10 plus years? Well, I hope we'll be here. Many thanks for your attention. It's really fun to share the journey with everybody.